Okay, so let's talk about a couple of concepts here. Evolution. What is it? We've talked a lot about it throughout the year. We've mentioned it. We all have some idea about what it is. We know it's change over time. A lot of people have different misconceptions about what it is. But at its most fundamental level, it's a change in allele frequency. Over time. So as the alleles in a population change their frequency, how many of any particular allele there are in a population, as they change over time, we call that evolution. That's it. It's not monkeys turning into humans. It's not alligators turning into butterflies. It's not any of the crazy ideas that Hollywood or anyone else would like to put forth. The consequences of this change in allele frequency over time is phenomenal. It has led to the evolution of species and the change of life on Earth. But I don't want to say subtle change. It's a subtle change. It's an additive change. These changes add up. So it's not just one event. It's not like the X-Men where they get irradiated by something and they change into something else. These are small changes that add up over time. So there's a lot that goes into this, a lot of ideas that go into this. Let's talk about the change in allele frequency randomly. So let's talk about some random change in delta, the Greek letter delta for change. Random change in allele frequency. What would that look like? So let's think about a population. A population's made of red creatures, green creatures, blue creatures, all of them living together, all one species, all intermixing, having babies. Maybe the red is dominant, maybe the blue is recessive, maybe they're not, maybe all three are co-dominant. Whatever happens, you have all these different variations. Now, this is a key point here. The differences in the marbles in this case, but in these little creatures, red, green, and blue creatures, it is genetic variation. Now, you've learned already that the genetic variation is due in part to their ability to have sex and their ability to mix up or recombinant DNA, right? Recombine their DNA. In meiosis 1, we get crossing over, which increases genetic variation even more, and then two individuals mixing their genes mixes up the genes even more. So you get a lot of, the diploid organisms in nature having sex so that you get this genetic variation of population. This is very powerful because having a large genetic variation, having individuals with different genes, is going to allow the population, the species, to survive. The more genetic variation any particular species has, the greater its chance of survival long term. So all these individuals are in this population. We call it a population of a species. By definition, a population is of one species. That's a group of organisms that live in a particular area that have babies with one another. We call that a population. So this is the parent generation. These are the parents. They're going to be living, eating, drinking, having babies. Something happens, the coronavirus or some other virus, something happens. It could be an earthquake, a predator or a parasite. But whatever it is, it's it's generally something with genetic drift, which is what we're talking about. Genetic drift is random, so it's going to be a random event. It's not going to select any particular individual. No one is more fit to survive than anyone else. It's just usually something like this is akin to a meteor hitting the island, a tsunami coming and wiping it. Very little warning, very little to do with anybody's individual traits. The fact that this is red and this is blue and this is green is irrelevant. The differences among them are irrelevant. There are differences, but the differences among them does not impact their survival in this example that we're going to call genetic drift. If their differences do not give them any advantage, then the changes in the frequency of alleles, and of course the changes of the frequency of alleles are represented by the color, right? Then the frequency of alleles, any change is due to random events. We call that genetic drift. So when we look at this and we see that this is called bottlenecking, right? So only some of these are going to survive. We don't know which ones, but if you pour these out of a bottle, you just happen to get mostly green and blues coming out, mostly green, some blues, and even less reds. Then in the next population, as these survivors, which by random chance the reds are mostly dead, then you see that in the next population you have mostly green and blue. This change in the population, I think you all agree it changed, right? It went from red, blue, green to just mostly green and some blue and a red. That change happened by chance. We call that genetic drift. I think that's pretty apt definition. Think of drifting randomly. Who knows where you're going to be in a year or two? There's no telling. It's all due to random events that you can't predict. So we call that genetic drift. Okay. This is not the only way that evolution occurs. In fact, it's just one of the big ones that happens. So let's take a look at, don't worry, you're going to hear all kinds of noises. My cats or my dogs or someone in the house doing something. No worries. So let's take a look at an example, real life example. You're at home. Your mom, your dad, maybe you don't like ladybugs or don't like these beetles, these bugs. So you get your fly swatter. Now these bugs are different. Some are red, some are blue. If you look here, 5 are blue, 5 are red. This is the genetic this is the ratio, right? This is a frequency. The frequency of reds is 5. The frequency of blues is 5. Their traits are directly related to their alleles, right? So when we talk about allele frequency, we're talking about the alleles that make the color of the bug the color that they are. So here you have 5 to 5, 5 reds, 5 blues. Something happens. You decide to smash them. You just swing. You're just swinging at the bugs. You're not trying to pick out any blue ones, or you just swing at the whole batch of them. And you happen to hit almost all blues. So now 4 of the blues are gone. Now you have 5 reds and 1 blue. 5 reds and 1 blue. All right, well, what does that mean? Who can have babies in this population? The reds and the 1 blue. So what's the likely outcome? What are you going to get mostly in the next generation? You're mostly going to get red. And you get red coming out in the next generation, only a couple blue, but mostly red. Now you got a 10 to 2 ratio after reproduction, after sex, after having babies. You have 10 to 2, 10 reds and 2 blues. So genetic drift is this idea that if you have genetic variation and something happens, randomly, you can get a random change in the population. This population, by the way, that's evolution. You just see, now you see evolution. So imagine that, these creatures here, uh, laid eggs but didn't have feathers. The red ones. The blue ones laid eggs but did have feathers. So you had a bunch of creatures that had, some had feathers, some didn't. They're all laying eggs, all having babies together. Some event, a meteor hit. Like, let's say they're the dinosaurs. Some dinosaurs had feathers, some didn't. A meteor hit. Those that had just randomly smashed a bunch of ones that didn't have feathers, and now you have a bunch of dinosaurs with feathers and some without. Could that have happened? Sure, that could have been an event that happened. It's not what happened, but the point is that this kind of change can lead to evolution, can lead to changing a population, seeing bluebirds turn red over time in many generations. Obviously, this can happen in 1 or 2 generations. The genetic drift can happen very quickly. So, although we talk about evolution happening over a very long time, the key is that evolution depends on genetic variation. You have to have a bunch of different genes for selection to occur. Now, genetic drift by definition has there is no natural selection in genetic drift. It's just random. Natural selection is going to select one of these traits to be superior, one of these traits to be an advantage. In genetic drift, the idea is that there is no advantage. It's random luck. We have to take in the random changes over time into account if we're going to look at the change in allele frequency over time, which is evolution. So when we're talking at the molecular level, they're generally speaking, you're talking about changes that occur mostly by genetic drift, and it usually is on a neutral mutations, right? So when you're talking about the inner workings of a clock, if you change the shape of a gear, generally the clock won't work. So only the changes that you make to still allow the clock to work will allow the clock to exist. If the clock doesn't work anymore, generally we throw it out, right? So the changes that happen to the molecules of a living organism have to be at worst neutral. Otherwise, the organism dies, and therefore can't have babies. So that mutation doesn't get passed on. So any mutations we see in a population must be at first neutral. So you have to have this genetic landscape, this population that has all these mutations. You have to have a lot of genetic variation, right? You have to have genetic variation, and if you have genetic variation, then whether it be genetic drift or natural selection, can take and impact the population over time. So now let's go ahead and look at an example and see if we can make some sense out of all this. So if we're looking at a mutation that's neutral, again, remember, mutation is neutral means it doesn't impact the function of the gene of the protein, so that the protein can still do its job, right? So there is no natural selection going to happen on this because it's neutral. That's what neutral means. It doesn't give it an advantage or a disadvantage. Some mutations are going to be a disadvantage, which means you're going to see less of them in the population. Some are going to be an advantage, which means you'll see more in the population because they'll be able to have more babies. I mean, that's kind of the idea there. But a neutral mutation is one that doesn't have an advantage or a disadvantage. So the rate at which a neutral mutation changes over time is fairly constant. Now, an advantage, a mutation that gives you an advantage, gives an individual advantage, is going to change in a population very quickly, because you're going to get many more babies that have that advantage in the next generation. That's the point of advantage. That's what advantage is. They're able to have more babies. However that works, you're going to see a really quick change in the population. That one that has a disadvantage, a mutation to one of these proteins that's a disadvantage, will also mean less babies, and therefore will decrease that gene's frequency over time in the population. So if the protein makes you slower or weaker, then you're not going to get selected for to have babies. So you're going to end up having less of your genes in the next generation. Where if you are the faster, stronger, handsomer, best able to provide, you're going to get selected for, you're going to have the most amount of babies. Over time, that's generally true. So that we see populations changing quickly with advantages and disadvantages and changes to the protein sequence. But if the protein sequence changes neutral, so it doesn't impact the function of the gene, doesn't give it an advantage or a disadvantage, then we see that these clocks, we can use them as clocks, because they're going to have anything that, remember with radiocarbon dating and with radioactive dating, radioisotope dating of rocks, anything that changes consistently over time we can use as a clock. So with molecules that evolve, that change over time, if we look at mutation rate, we can look at how often they mutate. And if they don't mutate very often, if they're conserved, right, which we've mentioned before, and the reason they'd be conserved is they're very, very important, and if any major changes or any changes that give an advantage that change the function of this molecule, it's likely that individual will not survive. Generally, something like cytochrome C, which is extremely important to carrying electrons in the electron transport chain, right? Cytochrome C is, you don't know this yet, but cytochrome C is an incredibly important protein to allow you to use the energy that you eat, that allow you to use the oxygen that you breathe in. Without cytochrome C, the whole system breaks down. It carries the electrons from the digestion of sugar over to make ATP for the cell. So when we look at how this protein's evolved over time, we can look at the simplest organisms around the yeast. We can look at their genetic code. We can look at humans and compare humans to all these. And again, what we would expect, just generally speaking, if you didn't know the answer to this, if you covered up the comparison of humans, pigs, chickens, dogfish, it's a fish, by the way, not a dog, Drosophila, wheat, and yeast, and you compared them, which of these would you think you would group together as being most closely related? Hopefully you say pig. Chicken, dogfish, a fly would probably be pretty far away. Wheat, a plant, it's not an animal. At least Drosophila is an animal, and fish are a plant. Wheat, and at least wheat is multicellular. Yeast is a single-celled fungi. So hopefully you would rank them in pretty much the order you see here as being most closely related to humans and least closely related to humans, without just looking at their structures, just looking at how they act and react. And so if this would work, then we would expect to see that the molecular data would support that idea, and in fact it does. But let's take a look at these positions. These are the positions. So this is the first position in the protein. This is the last position of the protein. We're going to look specifically at position 1, we can look at, well, they're counting here 6, but we'll look at position 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13 and 14, right? All right. I'm sorry. Positions 2, 3, 4, 5, 7, 8, 9, 11, 12, 13, 15, 16, 21, and 22. Those are where we see changes. So when we can go ahead and take a look at this and see how far back these changes have gone. And in the first position, there was no changes over time. We have exactly the same amino acid in the first position that yeast do, and wheat and Drosophila. So this is a protein, by the way. This is cytochrome C. This is a section of the protein, not the whole thing. So based on this idea that these mutations occur at a predictable rate, there's a specific amount of time, on average, obviously it's an average, on average these mutations occur at a specific amount of time. They're neutral, so they're not going to be selected for or selected against. If they happen, they're going to stay, so they're silent mutations, right? Well, they're not silent because we see the change in amino acid, but these changes in amino acids don't do anything to the function, so they don't give it an advantage or disadvantage. So it's like a silent mutation in the sense that it doesn't have an impact, but it's not a silent mutation because you do get a change in the amino acid sequence, so you must have gotten a change in the DNA that led to an amino acid change difference. So that being said, if there's 20 amino acid differences, these differences themselves can't tell us the evolutionary relationship among these. They are a clue. But if we look at the traits, at a bunch of traits, the fact that wheats are they do photosynthesis, they have chloroplasts, that yeast have, are single-celled organisms, that Drosophila is an insect that has a exoskeleton, that it lays eggs, that it has compound eyes. The dogfish has gills, is fins. A chicken has feathers and lays eggs. A pig has a placenta and gives milk and has hair. And a human, etc. All the characteristics. Then we can look at how long ago, and we find common ancestors between these organisms. We look and find in the fossil record how long ago that we had a common ancestor in the fossil record. And we compare morphologies and we compare DNA. So we can compare all that stuff, and that gives us some information as to the relationship between all these different organisms. And you have to consider all that, and scientists do look at all that when they're looking at relationships between organisms. But when we take a look at a molecular clock, when we take a look at 20 amino acid substitutions, if we had 20 amino acid substitutions between any two of these, and it took 400 million years ago, if the two organisms separated 400 million years ago, right? So the common ancestors 400 million years ago, and there were 20 changes in the amino acids. That means that those amino acids, on average, are going to change every 100 million years. 5 substitutions every 100 million years. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Here you're going to probably look at 200 million years ago. It took 200 million years for us to go from something that was more like a yeast for this protein, to something that's more like a human. So the sequence homology in cytochrome C, we can take that and we can organize all these organisms using that concept, and looking at phylogenies and looking at common characteristics. And we can start to say, well, we can predict that if humans and monkeys are so closely related, we would expect that there would be no more, where there would be a minimum of, 3 or 2 substitutions between human and monkey. So if we use this data, the differences in the substitutions in cytochrome C, just like we saw that this chart comparing these organisms, we can construct this tree. There's some common ancestors so very long ago, so with more than 30 substitutions, more than 30 amino acid differences in cytochrome C than we have in our cytochrome C. If we can actually construct a cladogram, right, a phylogenetic tree that that shows relationships between different organisms. So in this tree that's just based on the differences in the number of amino acid substitutions in the cytochrome C, we can see that we've grouped pigeons, ducks, chickens together, and that turtles are really closely related to chickens, but rattlesnakes are not. You see here, tuna is a fish, so that's even further back. The flyworm, the moth are connected together into a group. That also makes sense. They're insects. Yeast, the two yeasts are being grouped together using this process of just looking at just the amino acid differences in cytochrome C and how far back they would go. This process would group the yeast into one clade. So that makes sense since they're both yeast, they're both single-celled organisms, right? They do all the things that yeast do, they have all the characteristics of yeast. And it makes sense that the next most closely related organism is not a yeast but a mold, but they're all three are fungi, so that also makes sense. Again, the two insects being grouped together makes sense. The next closest the fact that turtles, chickens, ducks, and pigeons are grouped together, that makes sense. It's kind of strange the rattlesnake's out here, but okay. In this, we see that rabbits and kangaroos are grouped together, and pigs, donkey, horse, dog, monkey, and human. Now, in a general sense, it makes sense. In a general kind of broad stroke, it makes sense because all of these we would consider mammals, right? So kangaroos, rabbits, pigs, etc., are all mammals. The problem with this particular organization is that when we look at the differences in the physiologic, all the morphologic, the way things do things, the structures, kangaroos are marsupials, and according to this, the kangaroo would have broken off from a common ancestor to rabbit, and it would have marsupials come after placental mammals. But we know today that that's not true. We know that actually marsupials came before placental mammals. So marsupials were around before mammals that have placenta. So rabbit and kangaroo probably are not, even though this is correct as far as amino acid number differences between kangaroo and rabbit, the grouping of these two into one group is probably wrong, since this phylogenetic tree would have kangaroos deriving from a placental mammal. So it's probably incorrect. And how could this happen? Now, if this is such strong evidence, how come the differences between the amino acid differences between the two would group them together and would show a placental mammal coming from a mammal with a placenta, evolving from one with a placenta? The only reason we know that that's wrong is that we have a lot of other evidence to suggest the opposite. So when you're considering that, they're considering that question, the reason that this is wrong and the reason it can be wrong here, right, is and which brings into question a lot of these places, like why would you group these together? Even though a lot of these groupings make sense, horse and donkey, monkey and human, yeast and yeast, moth and fly, even though these groupings a lot of the groupings do make sense and do fit theory, some of these don't. And the reason that you have to consider is that these changes in amino acids are neutral. The number of changes the method that we're using as a molecular clock would suggest that something happened X number of millions of years ago. However, there's no real telling. You can't use that alone as your only source of information. If you do, you're going to have some error, and here are the errors. And the errors are going to stem from the fact that randomly you can have some event could increase the mutation rate or decrease the mutation rate, and you not be aware of what happened 100 million years ago. So using mutations, and even though these things are, maybe it wasn't genetic drift that changed it. Maybe there was an advantage to this amino acid being there 100 million years ago that isn't there today. There is no advantage today, but how do you know there wasn't an advantage 100 million years ago? So, because the conditions were different, right? It might have been warmer, might have been colder. There might have been a certain kind of food that allowed them to use that version of cytochrome C better than the others in the population. So there was no real way to tell 100. There are a lot of places where using this as a molecular clock would fall down. You can't use it alone. You have to use it in concert with other information, like morphology, like comparing what the common ancestors between placental mammals and mammals with placenta that carry the babies in their womb. That's what a placental mammal is. That has an umbilical cord and a belly button, right? Kangaroos don't have belly buttons because they don't have a placenta, because they're not placental animals. Mammals, they're mammals that came before the placental mammals that we evolved from. Mammals without placenta, mammals with placenta evolved from mammals without placenta. So, in any case, the point is that we have to combine all that information in order to come up with a much more accurate phylogenetic tree. And no matter what phylogenetic tree, no matter how much data you use, the only thing you can do is get closer and closer to the accurate result. You can't guarantee an accurate result unless you make a time machine and go back and actually see what happened over time, which we can't do. So all we can do is make really good guesses based on the evidence that we see in front of us. So when we look at questions like explain why chimpanzees and gorillas are considered most closely related to humans while monkeys are less so, well, hopefully you can list evidence like DNA sequences are more similar between gorillas and humans. Hopefully you can say something like there's less, and I don't know if this is true, but I would predict that there's less of amino acid sequence differences, less molecular, less amino acid sequence differences. There are morphologic differences. Morphologic means shapes and structures are greater between monkeys and humans than gorillas, right? The prime example is monkeys have tails, have long tails that they use for balance, while gorillas and humans don't. And there's other differences: brain size, jaw size, shape, etc., etc. There's a lot of differences and similarities and differences between humans and gorillas that don't exist between monkeys and humans. So when we're talking about highly conserved areas of the DNA, when you have a protein that's highly conserved, it's highly conserved because it's incredibly important to the function of the organism. Any major changes would probably not allow the individual to survive. So most of the changes that do occur are going to be neutral. So this is, in other words, have no advantage or disadvantage. In general, any change is going to be dangerous for the organism. So cytochrome C is highly conserved because it's an incredibly important function. And it also involves other proteins that it doesn't work alone, and so it because it works in conjunction with so many other proteins in a very complicated process. It requires that it has a specific shape to do a very specific function. So highly conserved proteins are good candidates for establishing protein homologies, because what you need to look at homologies, right, what you need when you're comparing two different species, and you want to look at what the differences between them is, you need something that's fairly stable, so that when you look at differences between species, it's not something that changes so much that you even have the same differences found within the same species. So you need something that changes, but changes rarely, so that there's plenty of time, so that when you see a difference between two populations, you know that it's not something you can group those changes, those different changes, into groups that we would call taxa today. So when you're looking at homologies, you're looking for things that you can use to help you group organisms, to be able to tell, well, this group of organisms are related and more closely related, or that group of organisms is less closely related. But in order to be able to say that, you have to be able to identify those changes as belonging to that group. And if it happens all over the place, you can't really assign the changes to a specific group. So highly conserved proteins are useful because they change rarely, and when they do change, we can identify those changes in the genetic record. Now, discuss some of the limitations using protein homology, specifically molecular clocks. Well, molecular clocks are not always that consistent, and that's kind of the problem. There's a lot of places where the question is, scientists still don't understand why the differences are as high as they are, or aren't as high as they would expect. In the case of the kangaroo here and the rabbit, why isn't the kangaroo? We know the kangaroo evolved is much further back. We know it did not evolve from a placental mammal. So why does it show so few changes or differences in in the molecular, in its amino acid sequence, where the rabbit, where it would be positioned here rather than back here somewhere. Well, it turns out that the different taxa, these molecular clocks are pretty consistent within a specific species, and it seems that different species have the clocks, the same protein, can change at a different rate within a specific species. Also, we're dealing with protein sequences, right? And so, because we're dealing with protein sequences, it's not it's not DNA, it's after replication. So you're going to have different changes in different species and different populations. So you could get epigenetic changes. In other words, you can turn certain genes on or off, or change how you express a protein, how you express a gene in a particular individual organism. So maybe you have more of the gene expressed, like you see in these three, or you have less of a gene expressed. That could influence these results as well. So the clock is not by itself the solution, but if you use it in concert with morphological, with fossil evidence, with physiological evidence, if you compare all this data, you could come up with a much more accurate cladogram, much more accurate family tree for living organisms. This might be a good point to ask, well, why is all this important? And I keep emphasizing why it's important, understanding. A lot of us are concerned about this COVID-19. Understanding where COVID-19 came from, where is it going, and how to treat it may all depend on our understanding of its genetic relationship with other viruses. And understanding that genetic relationship between other viruses could lead us to understanding how to treat it and how to come up with a vaccine against it. So understanding evolution, understanding these phylogenetic changes, understanding these genetic changes, understanding things like genetic drift, allows us to be able to come up with treatments, allows us to understand things like viruses and bacteria, and how to treat these things, and help us also predict how these things will evolve. For instance, antibiotic-resistant bacteria. They've been predicted, and they're coming out to be true. If we can predict how they would evolve, maybe we can prevent them from evolving that way. Maybe we can make changes in our behavior that would reduce the natural selection that's going on, so that they don't evolve the way we'd like. In fact, part of the success of us staying at home during this pandemic and stopping the spread of the virus is that we're going to, every time you spread it, every time it goes to a different host, there's a chance that it's going to mutate and evolve. And if we can reduce the rate at which that happens, we can come up with a vaccine that could eradicate it before it could turn into a yearly version of the COVID-19 infection, the way we see a yearly version of the flu today. So we could, using this technology, using this understanding of evolution, using this understanding of genetic drift and natural selection, we could come up with behaviors, rules, protocols that would allow us to prevent this virus from being a yearly occurrence and save millions of lives. All right, I hope this helped. Have a good day and keep safe.